COVID-19 therapy actually started many years ago based on this homology of some genetic and amino acid sequences with Ebola virus. You can see how the authorization process of this drug actually was in different countries. But it's necessary to stress that remdesivir could be applied only in clinics and only in intraperitoneal mode. So it's not peroral drugs. Another drug which probably could be associated with treatment of coronavirus disease is molnupiravir, which already approved by UK Health Authority. It's actually expected to be approved by FDA, but recent news actually, when the clinical trials were finished in more high-scale mode, it was shown that the promises for this drug is less than it was expected. So it's improved only 30% of the patients in their hospitalization and their health. Another new hope is Pfizer's novel Paxlovid drug, which actually combine this PF with some numbers ritonavir, which actually produce inhibition of some cytochromes and PGP. So in these clinical trials, it was shown that about 89% of patients have improvement in their health due to this first pill, which is still expected for the improvement. You can see actually overview of current and developed therapies in different reviews, and I just cite one review. Actually, in addition to remdesivir, molnupiravir, etc., you can see the monoclonal antibodies, convalescent plasma, fusion inhibitor, soluble ACE2, and other molecular decoys, etc., etc., etc. I will touch some of this in more detail later. Why repurposing of drug is so popular? Everyone knows that it's a long time to get a drug from idea to the clinics. But fortunately, in case of repurposing of drug for which safety is already proved, you can switch directly from the idea to phase 2 clinical trials. Of course, this is true if the drug is applied in the same administration mode and in the same dose. And of course, it provides the possibility to dramatically reduce the time, expenses, and increase the chances of success if you are able to repurpose the existing drugs. That's why the repurposing of drugs is a natural response on the COVID-19 pandemics. And when you look into the Google, you can see that more than 100,000 different documents are associated with drug repurposing against COVID-19. And first attempt of drug repurposing against COVID-19 combined in silico and in vitro methods was associated with dipyridamole. It was even in March 2020. And of course, dipyridamole is well-known antithrombotic, and it was shown that it probably could reduce the affinity of SARS-CoV-2 to its known receptor. So it could potentially combine the antithrombotic purposes with reducing affinity of SARS-CoV-2 to the human cells. But unfortunately, it was just first attempt, which was not confirmed in the further clinical trials. The next, more high-scale drug repurposing studies were published in April. And in first of this, 1,500 compounds were tested, about 13 were active at this cutoff. In another publication, over 5,000 compounds were tested, and 64 were shown active. But unfortunately, no correspondence between the results of this in vitro experiments. We have analyzed the available publications about 1 year ago and published paper in Pharmaceutical Chemistry Journal. You can see how people try to inhibit the replication of virus by existing drugs in different cell lines and the number of compounds that were tested. And some intersections between the results were shown for chloroquine, neflurine, remdesivir, nafamostat, etc. But of course, currently we know that chloroquine and neflurine are not efficient in clinics. You can see it in hydroxychloroquine as well. What are the current approaches to the drug repurposing? It's combined from in silico to in vitro in biochemical screening, cellular screening, and in case of drug repurposing, actually, you can do it directly in human in clinics. And of course, computational methods allow to make virtual screening of thousands and billions molecules. But in the top of this pyramid, you can see that just some small number of molecules could be tested. I already give this number in the previous slides. Here you can see how SARS-CoV-2 virus interacts with human cells and what are the induced pathological process. So based on this, some particular targets are suggested, from which most popular are main protease, RNA-dependent RNA, protease, TMPSS2, etc., etc., etc. And as I told you, in silico you can test many compounds, but in clinics just separate. That's reflected on this slide. The number of in silico significantly increase the number of testing in vitro, in vivo, and in clinics. It's important to stress that the number of clinical trials is much more than testing in vivo, in serum, hamsters, etc. That reflects the possibility for direct repurposing without testing in experimental animals. And you can also see that the most popular targets are main protease, papain-like protease, spike protein, ACE2, RDRP, TMPRSS2, etc. But main protease is most widely studied in this field. It's important to say that correlation between results that obtained in silico are not good in comparison with those obtained in vitro. Thus, you can see that it's necessary to be very careful by consideration of in silico results without experimental confirmation. And also, analysis shown that correlation of results obtained in different in vitro tests also is very poor. That's why that's actually caused by the absence of standard assays, a proven reference compound. And of course, due to this, results of in vitro tests also are not confirmed in further clinical trials in many cases. When you see how diverse are different in vitro methods, on example of SARS-CoV-2 MPRO, you can see very different materials that are used for study and very different methods of activity detection. Thus, it's not surprising that the results of tests obtained in different assays do not correspond each to the others. As I told already, SARS-CoV-2 main protease is one of the most popular targets for drug repurposing. That's also caused by the low mutation in this protein. Based on more than 5 million genomes in GSID database, you can see that this protein is one of the most slow mutated among other targets in comparison with some other like PLPRO, RDRP, or of course spike protein. What could be seen here, the most of mutations are on peripheral parts of this enzyme. So probably it's not change the active site of the main protease. But of course, one could not exclude some allosteric regulation or modulation when the conformation of the whole enzyme will change. And of course, even new stam like Omicron already caused some mutations in this protein as well. Actually, many people apply molecular docking to try to find which compound could be repurposed against SARS-CoV-2 main protease. One example was done by our colleagues, Veselovsky and others. In our institute, it was shown that neratinib is a potent inhibitor of this protein. But unfortunately, results were not confirmed by in vitro studies. Another example by other people is docking of nirmatrelvir into the binding site of SARS-CoV-2 major protease. This compound is produced as a drug by a pharma company in Russia for treatment hepatitis C. And in literature, one could see that nirmatrelvir inhibits this protease in vitro. So it could be considered as a candidate for repurposing. Another example that was docking provided by people from the Institute of Organic Chemistry, Zelinsky Institute. Here they showed that disulfiram and also neratinib could be potential inhibitors of main protease. For disulfiram, it was confirmed by in vitro studies. For neratinib, like I already showed for previous slides, it was not confirmed. I would like to mention also the hybrid in silico approach developed by Alexei Zakharov, our former PhD student, who is currently working in the National Center for Advancing Translational Sciences in CATS NIH. And they actually developed the workflow when they first tested some compounds in vitro, in cell assays, then use it as a training set, and then actually make the second stage for selected compounds based on the QSAR studies. And this increased the hit rate significantly. They found 3 new promising hematides, which were active also for some mutated variants of the virus. In the past year, in Europe, it was announced some joint European disruptive initiative, Grand Challenge against COVID-19. And we actually joined to this initiative. The terms and conditions was that it's necessary to make computational predictions by 3 independent methods for more than 1 billion compounds, including the launch drugs, at least for 3 targets from the 6 proposed by the organizers. We created the appropriate 1 billion plus library and actually selected 4 targets: main protease, papain-like protease, RNA-dependent RNA polymerase, and this serine protease, human protease. And I just show you the results using our proprietary software PASS and GUSAR, which were retrained on the newly collected training set. We selected some compounds with potential activity against those targets. And in general, in this challenge, over 100 teams took part, but only for a small number of them, results were selected for synthesis and testing. And our team actually was also shown here. But unfortunately, results of this testing are not available yet, but organizers say that preliminary results that are very promising. In the framework of our Russian Foundation of Basic Research project, we developed informational computational system anti-COVID-19, where we try to combine information collected from different sources and also some computational approaches to prediction for novel drugs. We think it may help people to filter the infodemics, because we actually try to select the most reliable information about disease, about biological process, about targets, and about pharmaceuticals which are either already used or are under investigation. And of course, it's possible to make prediction by using structural formula like it's in our PASS web server. So, for instance, for remdesivir, one can see that it has very high probability to be anti-coronavirus agent. Another point is that in case of SARS-CoV-2, we have very small number of compounds which could be proved as active against SARS-CoV-2. Therefore, we also added some possibility to analyze the similarity. And for instance, using molnupiravir molecule as a query, one could get some drugs which probably could be repositioned in this direction. Actually, we are deeply analyzing the literature data collected from different sources, from clinical trials, and take into account the drugs which are produced in Russia for repositioning. This is supported by Russian Ministry of Science and Higher Education. And actually, they will be tested against main protease in Chumakov Center in Moscow. It's necessary to stress that to treat COVID-19 patients, it's necessary not only treat the direct infection, but also treat the post-COVID-19 syndrome. And in addition to this, or maybe as a part of post-COVID syndrome, depression and anxiety is also important part. And in this field, recently, fluoxetine was shown as promising agents in some clinical trials. In general, a lot of publications about drug repurposing could reduce the chances to patent some drugs which really will be repurposed. That actually may reduce the interest of large companies to such repurposing campaign. And in recent review published with our colleagues from many countries, we posit that truly impactful computational tools must deliver actionable experimentally testable hypotheses, enable the discovery of novel drugs and drug combination, and that open science and rapid sharing of research results are critical to accelerate the development of novel, much therapeutics for COVID-19. Thus, to summarize my speech, I would like to say that drug repurposing is one of the current trends in discovery of more safety and effective pharmaceutical agents. It is the only way for immediate response on COVID-19 pandemics and other possible biogenic threats. Of course, cooperation between the research working in different fields of organic medicinal chemistry, pharmacology, virology, etc., is necessary to combat SARS-CoV-2, COVID-19. It's really multidisciplinary problem. And COVID-19 caused by SARS-CoV-2 is a complex pathological process. Its curation requires application of several drugs with different mechanism of action, taking into account the peculiarity of the patients. It's very important that COVID-19 actually is different for different patients. It's something like personalized therapy, which is not easy to apply by physicians who are working in hard conditions in the red zones. And the further fundamental biomedical research is needed to fill the lack of knowledge about the biology of the SARS-CoV-2 virus, which will increase the efficacy of countering this and new biogenic threats in the future. Thanks a lot for your kind attention. I'm open for questions and discussion. Thank you.
Thank you very much, Professor Poreyko. We had a fascinating lecture by you where you have highlighted that more than 1 billion compounds have been screened by in silico and then number of them by in vitro also. And over 100,000 publications have been made in this particular area. And all this has happened due to the active cooperation between academia and industry and the regulators, because never in history the regulator have reviewed the data as it is coming. So that is why we had the repurposed drugs coming out in 1 to 2 years, as you have mentioned, and the vaccines within a year. It is only because of that that the regulators have reviewed as the data is coming regularly, whereas otherwise they review the total data when it is generated, not the regular reviews, which has happened. And there was a great support from the academia to this exercise of the industry, and the industry's risks were overtaken by the government, because every government, wherever the vaccine or compound is being developed, they supported all the animal studies as also the clinical studies. All vaccines have been supported by that. Even in our country too, we have 7 vaccines which are some developed, one developed by our own country, that is the Covaxin, one coming from Russia, that is Sputnik 5, and one of the AstraZeneca, that is the Covishield, which is coming. And the number of others are in clinical trials at the moment and not yet approved. These are the 3 vaccines which have been approved. And the second vaccine from our country, there's a DNA vaccine, first time in the world, developed by the Cadila, that is Zacovir D, and that has been approved but not approved for mass use. They have found it to be effective clinically in adults as also in children, but not yet approved. Only 3 vaccines are approved in this country. One is Covaxin, the inactivated vaccine developed by India, per se, the AstraZeneca, that is Covishield, and the Sputnik 5 from Russia. These are the vaccines which are approved. And you have highlighted that 1,200 drugs were tested in silico and in vitro. They have shown activity in vitro, and you have, out of that, you have mentioned that remdesivir could see the light of the day. But remdesivir too, as far as the WHO trial is concerned, this was found to be effective. The confirmed claims only that the death time is reduced from 12 days to 10 days. That may be only the limited study they conducted in USA. But now this is FDA approved drug sold in that country and also sold in our country. The compound drug which costs 700 rupees to the company has been sold here for 1 lakh of rupees in the unofficial market. But in official market too, it is being sold at a cost of 20,000 rupees. Gileads have made billions of dollars from this drug, which is ineffective drug. And you have mentioned the favipiravir, omefiravir is found to be effective in our country in the trial conducted in Lucknow, but has not been seen the light as of the WHO trials are concerned. All have been found to be ineffective. Only remdesivir, they are continuing with that, maybe because of the industry high support with this. And the first which came from India was the hydroxychloroquine, and that was found to be ineffective in US as also by the WHO. So that is the state of the affairs as far as the total area is concerned. But I don't think pandemics come every alternate day. We were lucky in this because moment the first case was found in November 2019, in the first week of December, that is 10th December, the structure of the virus was known. And that is why everybody moved very fast and developed so many things. But neither in silico helped nor gene therapy helped. As you have mentioned, the knowledge of the pathology of the virus is still rudimentary. The drug, although remdesivir was developed for Ebola virus, you have mentioned, but in clinical trials by WHO, it was found to be ineffective. Only the trial conducted by Gilead in USA, it was effective. But no drug has started. As you have mentioned, the only ones which have molnupiravir of the Merck's, which has recently come, this too, as you have mentioned, is not effective. Paxlovid, we do not know of the Pfizer. At the moment, they are claiming the two Regeneron's antibodies, which came into fame when President of the US took it, previous President of the US took it, but they too are not, because the cost of each antibody, monoclonal antibody, is very costly. And as far as this area is concerned, I think this has shown to the world, if the academia industry combine together, they can do wonders. But support from the regulator, which can conduct a rolling review, which has never happened earlier in history. And this pandemic came after 100 years, that is after 1918 and 1920. That was before the World War I. And it is 100 years after that, this pandemic came. And maybe we don't see such type of pandemics in the near future. It took 100 years. And let us hope and pray that another 100 years are taken. But the variants of this virus which are coming now, Omicron is worrying the world over. Although we have found that Omicron, they are saying that it is more infectious, but less fatal. That's the story at the moment. And even though admissions in the hospitals are very less as compared to the Delta virus, which is around the world. And this Omicron is causing havoc in the UK as also in the USA. But luckily in this country, we so far have only less than 100 cases of Omicron. But add that to in all those people who has come from South Africa or different regions of Africa, and two came from UK, and they too have carried the virus to this. Luckily, we hope that it should not spread in this country. The symposium which happened in 2020, organized by JIPR, that happened just before the spread of the corona pandemic in this country. So we are lucky to all of us to visit JIPR and enjoy the hospitality of Dr. Saxena and his team over there and see the fine institution which has created JIPR. With these few words, I'll express deep thanks to Professor Proilikov, and I think we will have the discussion after all the three presentations are over. Thank you very much, Professor Proilikov.
Thank you.
We go to the next speaker. It is Professor Dr. Esen Akiyalon from Ankara University, Turkey. She is a fellow of the Asian Federation of Medicinal Chemistry. She is a medicinal chemist by profession, but is a chair in Ankara University in Department of Pharmaceutical Chemistry, more especially in medicinal chemistry, and she is in rational drugs design at that particular place. She has 100 papers to her and a book to her credit and a patent in for anti-cancer drugs. May I welcome Professor Yeltsin to kindly deliver her lecture.
Hi, everybody. Thank you very much for your nice, kind presentation, Professor Kambo. It is my pleasure to be together again with mostly the Indian people and my friends all around the world, which we know each other for many years. And I would like to congratulate Anil Saxena for the 7th year of his birthday. And as I know, he is very active and very creative scientist, and he had many contributions to the scientific world and still continuing. It is my pleasure to see his activities still. And Jaipur is also very happy to have Anil there. And hello also my friends, Vladimir, Atina, today's speakers and the tomorrow's speakers. So when they invite me, I would like to just talk about my activities, which I am interested in the cancer research and the DNA topoisomerase inhibitors for many years. So I'm going to just talk about one of my recent studies. Can you please enable me to just to screen sharing? I cannot do that. Can you give me permission to share my screen?
Organizers, please.
Okay. So how can I do that?
So they will be.
The screen has been changed. So what can I do? How can I do? Let me see.
You have opened your slides already on the computer and got minimized?
Okay.
Because you need to go back and then rejoin, open your slides first on the computer.
Yes, I opened already.
Minimized,
yes.
And then minimized them.
Minimize them. Okay. I do that.
Now you go to the share button and over there your slides will come and then open those slides. When you go to share button,
still it says that has disabled participant screen sharing. It says to me. So
that is Jipper organizers. Please permit her to do the screen sharing.
Yes, they should permit me. That's why.
Yeah. Jipper organizers, please.
Not
Professor
yet.
Nandi, permit her to share the screen.
Nobody hears me, I think. Okay.
Dr. Nandi, please permit your people to permit her to share the screen. Screen will not be shared unless you permit her.
You cannot start screen share while the other participant is sharing. So one of you who is sharing the screen has to close it.
Yeah. Previous speaker must have closed it.
I start. So can you please stop the last screen?
Professor Proikov has to close his screen. He would have.
Yeah, maybe. Yeah, maybe.
Yeah. Unless he closes, you can't share.
I cannot yet.
Yeah. Professor Proikov, organizers, please see that the previous screen is open or closed. Previous presentation.
I cannot still.
Yeah, I too am requesting them. I don't know whether they are able to, they should be able to hear me and do the needful.
Let me see. Okay, now. Okay. It's okay,
Okay,
now.
okay, please.
All right.
Yeah.
Okay. If I go to the slideshow, can you still see my slides?
Yeah.
Okay.
You will start.
Okay. Thank you very much. Okay, I'm going to just talk about the insight into mechanism of action of anti-cancer heterocyclic compounds. And let me just show you some pictures from my faculty. Can you see the pictures of my faculty now?
Not yet. Your screen has not moved.
Okay, I think if I go to the screen share, then I think it is impossible to just move the slides. So I can go on maybe like that. Okay. So then it's changing now.
Yeah. Yes, it has.
Okay.
Yeah.
Okay. If I screen share all the slideshow, I think it doesn't move. All right. Okay. This is a picture inside of my faculty from the Ankara University, and my office is over there. And there is another picture at Dean's office here. It's another picture inside of the faculty. This is the laboratory of ours, the Faculty of Pharmacy, Ankara University, which is in Ankara, at the capital of Turkey. And this is our research laboratory of pharmaceutical chemistry, a picture from that laboratory. And ladies are just working there. And now I'm going to just talk about the heterocyclic systems as benzozoles. And we have been many years working on this kind of compounds. And benzimdazoles and related fused heterocycles, such as benzoxazole, benzotiazole, are the structural isomers of natural nucleotides, like adenine and guanine, that allow them to interact easily with the biopolymers. These isosteric heterocyclic compounds integrated potential activities as anti-tumor, antiviral, antibiotic activities as the new topoisomerase 1 poisons, and HIV-1 reverse transcriptase inhibitors and DNAGRase inhibitors. So they have lower toxicities while we just work on that compounds for many years. And now I'm going to just talk about the DNA topoisomerase 2 inhibitory activity of our synthesized compounds. These DNA topoisomerase 2 is in the cancer development takes place, and the inhibition of this enzyme can cause the anti-cancer activity. Then eukaryotic topoisomerase 2 enzymes are essential for several cellular processes: replication of DNA, transcription of DNA, chromosome condensation. Topoisomerase are the enzymes that control and also modify the topological states of DNA. They can catalyze several interconversions between topological isomers of DNA by transiently breaking single or double strands and resealing them after reorganizing on the topology. And the structure of the eukaryotic DNA topo2 alpha is you see here. There is the twofold axis of the symmetry aligned vertically between subunits. And this is the N-terminal region, and this is the DNA binding cleavage domains, and the catalytic regions of the subunits you see here. And DNA topoisomerase, how it acts, it is called as the two-gate mechanism. Topo2 binds one strand of DNA. If I can do it in a slideshow, maybe you can see what happens in that way. Can you see? Topo2 binds one strand of DNA, cleaves it, passes another strand through it, and then seals the two cleavage ends back together. And this is the active site of the enzyme. So I go to the normal. Here, there is the publication. In early years, we worked with that topoisomerase tool. And we synthesize the compounds as the inhibitors, having benzoxazole, benzotiazole, benzimidazole, benzoxazole derivatives. We mainly put some substitutes all around. And because we know that at the second position, it is responsible from the activity itself, and these groups have contributions by increasing or decreasing their activities, and we choose many physicochemically different substituents all around, and we put some bridges over here, and we also use some heterocyclic systems attached to that benzozole ring. So in our laboratory, first we do the mainly the synthesizing work, and then go into first deciding which compound to synthesize. We use molecular modeling studies, and afterwards we synthesize the compounds and test their activities, and try to just put some conclusions about regarding their mechanism of action, and we tested their compounds, our compounds, against topoisomerase 2. Toposide is a main standard drug used as topo2 inhibitors, and it has the activity as IC50 values as 21.8. And our compounds, as you look at their activities, some of them is more active, and I don't take the non-active ones or not having good activities. And then we just tried lead to hit as the structure-based molecular modeling approaches. We used homology modeling, molecular docking studies, sorry, complex-based pharmacophore generation, and virtual screening later. So to just find out some other drugs which will show the same activity. Now, for the homology modeling studies, we first we used Drosophila DNA topoisomerase 2, and there is no any crystal structure of this enzyme in PDBs. So the sequence of this enzyme obtained from UniProt. Modeler software, which is provided in Discovery Studio, is also used in our study. And the templates were found for the sequence using BLAST search. The sequences of the top prim domain of human topoisomerase 2 alpha and beta are 89 and 88 similarity to the Drosophila melanogaster. We choose two human topoisomerases, 2 alpha and beta, for that reason, and carried our work with the second one as the human topoisomerase. We did some docking studies. We compare both induced fit and gold docking studies. Induced fit, we used Maestro Schrödinger's program software and Gold from the Discovery Studio. We found out their activities according to these softwares and compare their results with each other. In the induced fit docking studies, standard virtual docking studies assume a rigid receptor, but in reality, many receptors alter their binding site to conform the shape and the binding mode of the ligand. And this is often referred to as induced fit. Schrödinger's software provides an induced fit docking, so we used it. In the induced fit docking studies, we superimpose the duct etoposite from the human DNA topoisomerase. We just get from the human topoisomerase, which is duct, the etoposite duct in. And after the induced fit docking and the ligand taken from the homology model, and we take our ligand from the homology model and then superimpose them, the RMSD value is 0.67, as you see here. These are the most active compounds. You just see here some of them, which is comparable to etoposite's activity, like 24 or 17 or 11, like that, the IC50 values. And then we did the induced fit docking, and then we see that the ligand is reacting with the enzyme interactions and DNA interactions both. In the enzyme interactions, we mostly see arginine 523 is very important because we see it in every of them. And if there is no any interaction with the enzyme part, then there is no activity, as you see here. This is a good finding of the work. And the inhibitor binds to the transient enzyme DNA complex and inhibits the relegation of DNA, generating single and double stranded breaks that harm the integrity of the genome. So this ultimately leads to the accumulation of DNA strand breaks and also cause the cell death. And this is one of the docking study of the homology model, which we did. And there is another one, as you see, is one of the best activities in the binding site of the homology model. And this is the 2D ligand interaction diagram of 1A from induced fit docking studies. And now we come to the gold docking studies. Binding site was defined from 5 angstrom around the ligand, and the binding site residues were kept flexible during docking procedure for comparison to induced fit docking method. And best pose of the largest cluster was chosen for each ligand. Then the structural superimpositions of the duct etoposite, purple, as you see here, at human DNA topoisomerase, and the ligand taken from the homology model. And the RMSD is also valid, and the superimposition is nice in that study. And how it reacts, what are the interactions? At the gold docking studies, you see here arginine 523, as I told you before, the same amino acid, and etoposite is a huge molecule. This is the etoposite interacting with the receptor. It is a huge molecule, and the cleavage site of the enzyme is widely opened for that reason. And for that reason, the binding site residues kept flexible. And now we come to the enzyme interactions and DNA interactions. There is both with the enzyme interaction with the DNA interactions, and some of them which has no any activity, has no any interaction with the enzyme. And arginine 523 is also important in this docking studies. So afterwards, we refined the docking results. So what we did, then we start with the first step, energy minimization of the hydrogen atoms using a fixed atom constraint to all other atoms of both protein and the ligand. And the second step, the minimization of protein side chains and the ligand atoms while the protein backbone kept fixed. And the third step is the minimization of the residues, 5 angstrom around the ligand while the other parts of protein kept fixed. So 1A, in the active site of the homology model of Drosophila topoisomerase, after refining, we see the same kind of interactions with DNA and also with the enzyme site. And 3F, we see the same kind of results, same kind of interactions here. Then the docking studies with human type topoisomerase, we studied with it. The binding site residues were kept flexible during the docking procedure. Purple one is the docked etoposite, purple, and they obtained from the homology model. We just superimposed them as the good RMSD value. And here are the interactions after refining. The 3F enzyme at the DNA cleavage site of the homology model, and with the human topoisomerase 2 beta enzyme. So we see the same kind of interactions after the refinement, and you can see here compound 1A at the enzyme DNA cleavage site of the homology model, and in the human topoisomerase 2 beta. So these are the arginine 503 and arginine 523 resembles each other in the same position. When we compare the results of these studies used with two computer techniques, gold has better correlation than induced with, we found out. And also gold is a much faster method than induced with docking. So complex-based pharmacophore studies, the most active compound 3F and 1A were chosen to carry out complex-based pharmacophore studies. Complex-based pharmacophore models were generated from the selected refined complexes obtained both from gold docking and induced with docking studies. And we use the ligand scout for point 2. Here are the complex-based pharmacophore generation with the compounds using gold glide, the duct pose of the 3F, and also we choose 1A, which are the most active ones to create the pharmacophores, to generate the pharmacophores. And we found out 4 pharmacophores. Why we did these pharmacophores? So we would like to virtual screening afterwards with the other compounds getting from the databases. And so we have 4 pharmacophore models. You see here as the yellow spurs the hydrophobic features, and here hydrogen bond acceptor areas, and the blue rings are the aromatic ring features, and the gray sphere excluded volumes over there. And after the virtual screening, the compound database of the National Cancer Institute contains a collection of about 250,000 compounds. So we used it. They mostly tested in vitro for statoxicity against the human tumor cell lines. and to retrieve the novel TOPA2 inhibitors and potential anti-cancer agents. The pharmacophore models are applied sequentially in virtual screening of the NCI databases. And according to the pharmacophore 1, we found out 1,530 hits. And when we come to the pharmacophore 2, we found out over 1,000 hits in that way. And with the pharmacophore 3, we found out more than 16,000 hits. And afterwards, we just tried with the pharmacophore 4. And there are 65 hits here again. So after these findings, we filtered them by the Lipinski screening and to find the common hits. No common hits were screened along with the 4 pharmacophore models, and 3 compounds only screened both pharmacophore 1 and 2, and other 42 compounds were screened both pharmacophore 1 and 3. Then, after all of the studies, we choose 15 hits as promising for evaluating their gold score values, interaction with the protein and as well with the DNA. And one of the compounds is a potential intercalator, so it was eliminated from the work. And then we come out with the other 14 compounds. You can see here their structures, which are some of them similar to our compounds. Some of them have some differences, but they just fit into that pharmacophores. And here are the other 6 compounds you can see here in that screen. And then we did their interactions. We tried out their interactions with the gold score, having good gold scores. They have hydrogen bond charge, PP interactions, P charge interactions, and hydrophobic interactions. So we just studied with them. And as a conclusion, I can say that topoisomerase 2 enzyme is an important target for the anti-cancer research. 2 docking methods, gold and induced fit, were also compared in the study. Due to the studies, the selected compounds from NCI database are worthy to carry on the anti-cancer in vitro studies. So it can be concluded that after biological tests completed, this research also provides a model to design a novel or potent anti-tumor agents as human topoisomerase 2 inhibitors. So this is the recent publications of our study in the current topics in medicinal chemistry in 2020. And the achievement we applied for the patent of the most active compounds, and we had recently good results on the animal test, and we hope to carry on our studies in the clinics. So this is the website of our research team, www.ssresearch.org. So you can find every kind of our publications and every kind of activities we have done here. And you see pictures from my research team. In that search, we also have the collaborator from the Sydney University, Australia, Renata Griffith, as most of you know. And in our faculty team, you know, Professor Yalchin is my colleague. And we have these kind of PhD students. Some of them is changing now, but we have the more recent photo from our group. And also for their biological tests, we did the synthesized and the molecular modeling studies. And for the biological tests, we have from the Hacettepe University, Nuran Dirir and her group. So this is all my talk. This is my talk, and this is a lovely picture from Istanbul. If you haven't visited yet, please come to visit. I am in Ankara at the capital, but Istanbul, as most of you know, is the well-known city in the world. So thank you for your listening. And if you have questions, I would like to answer them.
Thank you very much, Professor Hassan. Akli Yeltsin, you showed beautiful pictures of your department and now also of Istanbul. One is lured to visit the university as also the city. Very nice pictures.
Yeah.
She has taken us to a panoramic view of the heterocyclic benzoxazoles, benzothiazoles as anti-cancer compounds. She has done all types of jugglery which the chemist does using bioinformatics as also the QSR studies to develop as also the pharmacophores and the inhibitory enzymes. Topoisomerase 2 of the human, she has found that some compounds are showing excellent activity against this, and she has found also 4 to be active in animal studies, and we hope and pray that one of them becomes an anti-cancer drug in the near future.
Yeah, we hope
Thank
so.
you very much. Yeah, thank you very much, Professor Aysin.
Thank you too.
Yeltsin. Thank you.
Thank you too.
Thank you very much. We'll have discussion at the end of the 3 talks. Now the next talk is by Professor. Yeah, the next talk is by Professor Giro Niki Kaki.
Hi, Athena.
Hi, Aysin. Yeah,
Athena,
great.
yes, yes.
Yeah, it's very nice to see you here.
Me too. At least by screen, if we cannot see each other.
Okay.
He is PhD in chemistry and professor and head of the Department of Pharmaceutical Chemistry and vice president of the School of Pharmacy. A member of the Mediterranean and European Academies of Science and Arts, School of Pharmacy, Aristotle University, France, Greece, Thessaloniki City, Greece. Welcome, Professor Giro Nikiki. Kindly initiate your talk. On thiazolacrylamines as antimicrobial agents.
Dear Chairman, dear guest, first of all, I would like to congratulate and say to my old friend, more than 20 years, Anil Saxena, happy birthday. I would like to wish him good health, continue to be active in his science, and all the best for him and his family. Here, I just want to remind to Anil some nice moments which we spent together in different time of our scientific and private life. I hope that Anil remember this. It was a trip in the Kashipur, when we came to the Kashipur for the first time. Here we are in Kotor. Here we are in conference in Moscow. And here again we are in Kotor. So now coming to my talk, first of all, I would like to tell you that maybe I will disappoint you because I am not a computational chemist, I am a medicinal chemist and just use computational tools for my research. My talk will be, it is dealing with the antimicrobial agents in silico and in vitro synthesis, in silico and in vitro evaluation. So during my talk, I will point out one introduction, design and synthesis, biological evaluation, and conclusion. So throughout the course of human history, there was an ongoing battle between the human people and the variety of microorganisms that cause infections and diseases. In the past, many diseases such as swine appeared in form of pandemic. From this battle, winner was a man, and some of them, like the plague, disappeared. Unfortunately, there are new infections and diseases affecting large human population, causing significant morbidity and mortality. One example is acute immunodeficiency syndrome, and recently we have another very dangerous infections, which is SARS-CoV-2, which during 2 years take a lot of human lives. So why it's important to create antimicrobial agents? As you see here, the bacterial infections affect all our organs, all human body, and sometimes most of them
slides are not
very
moving. Slides are not moving. Slides, we see the first slide only.
I don't know. It doesn't move?
No, it doesn't move. It's on the first slide.
Are you on the first slide or you have moved? Have you gone to the second slide?
I don't know what is going on.
First slide is visible.
Now it's moved?
Now it's moving.
Yeah.
Moving, okay. So, but the relationship with microorganisms in humans sometimes may be beneficial, as in the case of non-pathogenic microorganisms which inhibit the gastrointestinal tract and contribute to the functioning of the digestion of food and production of vitamins, and sometimes are harmful. Diseases caused by the pathogenic microorganisms classified as infectious. And as you see, all these infections sometimes become very severe and leading to the fatal to the death. Since their discovery and during the 20th century, antimicrobial agents have substantially reduced the threat posed by infectious disease. But these gains now seriously jeopardized by another recent development, this emergence and spread of microbes that are resistant to cheap and the effective first choice of first-line drugs. The bacterial infections, which contribute mostly to the human disease, are also those in which emergent and microbial resistance is most evident. From another hand, the economical crisis, high cost of industrialized medicine, insufficient public access to medical and pharmaceutical care enhance the rate of the infectious disease, which become a global problem. They have monopolized for centuries the factors of death and disability of a million of humans, and presently plaguing and even ravaging the worldwide each year with a far surprising performance than war. The growing challenge on the health of human progress posed by infectious disease, aggravated by the continuous emergence of new and old endemic infections of global impact. During the past 2 decades, the world scientific community has besieged by tremendous concerns caused by diseases whose incidence in human has augmented the reason, such as the emergence of new pathogens, development and of antimicrobial resistance. At least 30 new infections have been insidiously scattered to threaten the health of billions of humans across the planet, especially in low-income countries. Unfortunately, for many of them, there is no efficient treatment or vaccine alongside with the limited scope of control or prevention strategies. So the treatment of infectious disease still remains an important and challenging problem because of combination of factors, including emerging infectious diseases and the increasing number of multi-drug resistant microbial pathogens with particular relevance to gram-positive bacteria. You see here the diagram of growing resistance of bacteria. And now in 2020, another bacteria, which is vancomycin, it's appeared a resistance. The action of known antimicrobial drugs deals with inhibition of limited spectrum of cellular processes, such as biosynthesis of protein, DNA, cell wall, and folic acid. Despite the continuous success in creating inhibitors against such targets, the frequency is low comparing to the period of golden era of antibiotic drug discovery. Moreover, pathogenic bacteria have developed sophisticated intrinsic drug-resistant mechanisms that are hard agitated in the microbial metagenome as a natural phenomena, and thus it's very difficult to treat. The rates of morbidity and mortality, mainly due to the low effectiveness of available medication and the development of resistant strain, rose up. The problem became more serious in case of immunocompromised patients who are more disposed to the opportunistic fungal infections. The treatment of such infections is quite difficult due to the spread of antifungal drugs resistance, generally in patients who often are subjected to the antimitotic therapy, and especially to immunocompromised patients suffering from cancer, age, and organ transplants. So, to overcome a potential approach to overcome the resistance problem is to design innovative agents with different mode of action, so no cross resistance with the present therapeutic can occur. A synthetic linezolid, a synthetic molecule of oxazolidinone class, has been approved for human use against the gram-positive bacteria and appear to have unique mechanism of antibacterial activity. So, the main targets of antimicrobial drugs are cell walls, synthesis of RNA, synthesis of DNA, and metabolic pathways. Here you see the drugs which worked on each of these targets. As already mentioned, resistance is a growing problem, and it can be endogenous or physical or secondary and acute. And mechanism of resistance is threefold: modification of target receptor, inability of the antibacterial to enter the cell, and the activation of antibacterial. Here you see all these mechanisms, such as alteration of the drug target, inactivation drug by enzymes, and so on. The question is why the bacteria have so high degree of resistance. And probably the answer is that they multiply rapidly. They have the ability to exchange between the genes. And the third reason is that there is the lack of efficient and very good antimicrobial with different mode of action. Actually, only 2 such drugs have been approved during these last 30 years by FDA. It is linezolid and daptomycin. From epidemiological point of view, the resistance through the plasmid is thought to be very important. As I already mentioned, microorganisms and mainly bacteria are able to exchange their genetic material in 3 main ways: conjugation, transformation, and transduction. And thus, it is possible to acquire new genes that, which are often responsible for the occurrence of the resistance. As in case of the bacteria, and fungi too work with the same, have almost the same targets. And as bacteria, fungi too developed resistance, so there is a need also for the creation of new antifungal agents also. So, taking all this into account, the aim of this project is to find structure of compounds that can be effective antimicrobial agents. It is known that indole derivatives attract the interest of scientists due to their wide range of biological activities. As you see, it is only some examples. They can be they can pose antimicrobial, anti-inflammatory, analgesic, COX, anti-cancer, and so on, anti-HIV. From another side, this indole moiety was found in exists in many approved by FDA drugs for human use. This is indomethacin, anti-inflammatory drug agent, yohibin, agent approved for the sexual disorders, and delaverdine, anti-HIV drug, and reserpine, which is known antihypertensive agent. On the other hand, it is known that natural products have served as a power source in the drug discovery. There have been many useful, important drugs developed from plants and microorganisms. One of them is streptochlorine, which was its bioactive compound with antifungal activity, firstly isolated from the marine streptomyces by Japanese scientist Watanabe. Another interesting core is thiazole, which is also, according to the literature, have a wide range of biological activities such as antimicrobial, carbonic anhydrase, analgesic, antiviral, anti-HIV, anti-diabetic, and so on. This core, as in I'm sorry, I don't know what's happened. Just a moment. I will
He's
see.
unable to move the slides. Organize this, please.
Okay. The thiazole ring is also present in many approved drugs. It is penicillin derivatives, bleomycin, thiazophorin, anti-neoplastic drug, vitamin B1, thiamine, ritonavir, anti-HIV drug, and meloxicam, anti-inflammatory agent. So, we based our design on the one of approaches in drug design, which is molecular hybridization, which is based on combination of two or more pharmacophores of different biological active molecules in the frame of one single molecule. The aim of this approach is mainly to improve the activity profile and reduce the unwanted side effects. So we combine in frame of one molecule to these active pharmacophores, indole and thiazole. After the design, we went to the synthesis of this compound and substituted 3 alpha chloracyl indoles obtained by acylation of corresponding indoles. They are used as initial compounds, and after reacting with the urea in methanol in the presence of base, they give the target compound for indole 3-il thiazole 2 amines, which you see here. Acylamines we synthesize starting from already synthesized thiazolamine by reacting with the acid chlorides of the corresponding acid in the presence of pyridine. Totally 29 compounds we synthesized, say 16 are indole derivatives and 13 are methyl indole derivatives. Next, we make evaluation of antibacterial and antifungal activities, and evaluation of these activities was performed using microdilution method. Minimum inhibitory, minimum bactericidal, and minimum fungicidal concentration we determined using as reference drugs the ampicillin and streptomycin, and for antibacterial and ketoconazole and bifonazole. I'm sorry, a panel of 4 gram positive, 4 gram negative, and 6 fungi were used for this evaluation. Here we see the results of antibacterial activity of indole-based thiazole derivatives, and as you can see, many the best activity was achieved for compound 5X, followed by compound 5D and 5M, but all these compounds showed very good activity, mostly comparable and even higher than reference drugs against some bacterial species. Here you see the structure of the 3 most active compounds and their antibacterial activity. The 3 most active compounds also were tested against 3 resistant strains: methicillin-resistant Streptococcus aureus, Pseudomonas aeruginosa, and Escherichia coli. As you see from the graphs and the table, all compounds appear to be more active against methicillin-resistant Streptococcus aureus than reference compound ampicillin and even streptomycin because it doesn't have any MBC minimum bactericidal activity. These three compounds also we tested for their ability to stop the biofilm formation, and obtained results are promising because these two compounds, 5M and 5X, in concentration of MIG, are able to stop biofilm formation with higher potency than streptomycin and ampicillin. Compound 5X was active in lower concentration, 0.47, compared to 5M, which was potent in 0.84 milligram per ml, but it should be mentioned that this compound appear to be more potent than reference drugs, even in concentration of half MIG. So these both compounds have displayed strong antimicrobial potential, represented by both MIG represented by low mix towards the non-resistant and resistant bacteria, accompanied by strong anti-biofilm potential towards Pseudomonas aeruginosa. Since majority of infections are associated with the development of with the biofilm forming microorganism, these two compounds are promising, have promising potential for the development of novel anti-biofilm therapeutics, since they reduce the growth of both planktonic and biofilm-associated microbial cells. These three compounds also we studied for their these three selected compounds we determined for their interaction with the antibiotic streptomycin, and the results showed that there is additive effect, which is taking into account the in vitro data. The combination of this compound with the antibiotic can reduce MIG and so subsequently increase their efficiency. The bactericidal nature of these three compounds was studied against Pseudomonas aeruginosa colonies by time kills curve assay. And it was found that all these three compounds, after 1 hour of treatment, reduced the number of bacterial CPU till up to 90%. While after 2 hours, they reduced it up to 94%. After 6 hours, no one of Pseudomonas aeruginosa colonies treated by these selected compounds remained viable. Here we see the antibacterial activity of methylindole-based thiazole derivatives, and as you see, they appear to be less potent, and only some of them showed moderate activity against some of the bacterial strain. Antifungal activity of indole derivatives here also showed that the best activity was observed for compound 5X, followed by 5D and 5M. And again, all these compounds showed very good activity, higher than both reference drug, ketoconazole and bufonazole. At least these compounds in red showed activity higher than all that both reference drugs against some bacterial species. And here you see again the results of this. Antifungal activity of methylindole derivatives was evaluated and show two good activity. In red, in light red, it is compounds which are more potent than reference drug. In maroon, it's compounds which show the activity comparable with reference drugs. So you see that actually the compounds are quite active. And here you see the structure of two more active methylindole derivatives and their activity. In order to estimate the probable mechanism of the antibacterial action, all the tested compounds we studied For all tested compounds, molecular docking studies were performed on different bacterial targets. DNA topo4, Escherichia coli primas, GRAS, thymidylate kinase, and Escherichia coli murbi. From this table, it is obvious that the calculated free energy of binding of Escherichia coli to Escherichia coli murbi was the best one comparing with all other targets. So it may be concluded that the inhibition of Escherichia coli murbi enzyme is probably the most suitable antibacterial mechanism of action of these compounds. The most active compound, 5-He, exhibit the lowest binding energy of minus 10.74 kilocalorie, forming 3 hydrogen bonds. One among between the hydrogen of the indole ring and serine, oxygen of serine 228. This hydrogen with distance 279. This bond is very important because this residue, serine residue, is take parts in the proton transfer of at the second stage of peptidoglycan synthesis. Also, it forms another 2 hydrogen bond between the hydrogen atom of the amino group of the side chain and oxygen of asparagine 50 and glutamate 324 with distance 292 and 247, respectively. The compound is directed into the Escherichia coli murbi active site, enzyme active site, in the channel composed of amino acid isoleucine 121, proline 110, arginine 158, glutamate glycine 122, glutamate 119, arginine 326, with which it forms a hydrophobic interaction, which all these contribute also to the stabilization of complex between enzyme and compound. The molecular, here you see the results of molecular docking free binding energy to the antifungal targets, the hydrofolate reductase and KIP51. And as you see, KIP51 seems to have the best energies, and probably it is the main mechanism of antifungal activity of these compounds. Compound 5X showed the lowest, first of all, docking result studies revealed that all these compounds may bind to KIP51 enzyme in the analogous way that do ketoconazole, the reference drug ketoconazole. Compound 5X showed the lowest binding energy of minus 1255 kilocalorie per mole, and this is due to the hydrogen bond formation between the nitrogen of the indole ring and hem group of the enzyme. Also, there is plenty of hydrophobic interaction with phenyl 380, phenylalanine 233, valine 509, methionine 508, tryptophan 311, phenylalanine 228, phenylalanine 126, tyrosine 132, and all these contributed to stabilization of complex enzyme compound. Furthermore, compound 5D is placed in enzyme and in the enzyme by the site of hem, forming by binding with hem by hydrophobic and by aromatic interactions. And this the interaction with the ferrum is is more strong than interaction of that hydrogen bond. So these compounds, that's why these compounds, both 5D and 5X, are more strong than ketoconazole. Ketoconazole, the benzene ring of ketoconazole also form the hydrophobic interaction with hem 601, but the hydrogen bonds and bonds with the ferrum is more strong than hydrophobic interaction with hem. Here the data of the methyl indole derivatives. Again, for methyl indole derivatives, the most putative mechanism is probably inhibition of KIP51 of Candida albicans enzyme. The most active compound 5T showed the low binding energy, minus 1128 kilocalorie, and this is due to the formation of hydrogen bond with enzyme and ferrum of hem, and also another hydrogen bond with the nitrogen of the side chain of the compound and hem. And also here you see plenty of hydrophobic interaction of from tyrosine 132, tyrosine 118, isoleucine 131, phenylalanine 126, leucine 376, and tryptophan 311, which contribute to complex stabilization. The same we see for the compound 5R, which is also placed in from the side of the enzyme, from the side of ferrum of the enzyme, and form interactions with ferrum of the hem. In order to see whether our compounds can be bioactive and oral drug candidate, the prediction of drug likeness was performed based on several rules, such as Lipinski, Ghosh, Weber, Egan, and Moeg. And as you see from the table, no one of compounds show any violation from these rules, and show the bioavailability score was about 0.51. Moreover, all compounds showed moderate to good drug likeness score from minus 63 to minus 29, which was the best predicted drug likeness score. And here you see the bioavailability radar of the tested compounds. The established human fibroblast-derived MRCC5 normal cell line were chosen to be applied in this work because in order to test the cytotoxicity of the newly synthesized chemical molecules, since this application has been licensed by the regulatory bodies for human vaccine and new drug development. So the MRCC5 was used cells. They are used in two different concentrations of 10-5 and 10-6 for the evaluation of cytotoxicity of the new synthesized compounds. No substantial effect on cell proliferation after 48 hours exposure has been observed in the cultures, since the growth was equal to 80 or higher of 80 for all tested compounds compared to the control untreated cells. Moreover, the percentage of dead cells accumulated in the culture was very low, since its maximum doesn't exceed 2 or 2.5%. Coming to the end of my talk, I can conclude that the subgroup of indole-based thiazole derivative showed good antibacterial activity. The most sensitive bacteria was Salmonella typhimurium, and the Streptococcus aureus was the most resistant. The most active compound, 5D, 5X, and 5M, were active against resistant strain MRSA, Escherichia coli, and Pseudomonas aeruginosa, showing better activity against MRSA than both reference drug. Two compounds, 5M and 5D, were able to stop the biofilm formation, being more potent than both reference drug, even in concentration of 0.5 mg. And all three compounds showed additive effect with streptomycin. Activity of compounds was found to depend on the nature of substituents and their position in indole and thiazole ring. All tested compounds showed good antifungal activity against all fungi tested. And docking study, prediction of mechanism of action by docking, revealed that MUR-B inhibition may be a putative mechanism of their antibacterial activity, while the inhibition of 14-alpha demethylase may be the responsible for their antifungal action. Tested compounds are promising multi-target agents against the bacteria and fungi, and compound 5X can be considered as a lead compound for the development of new antimicrobials. And before closing my talk, I would like to thank all my collaborators who were involved in this project, and By the end, I would like to show you some pictures of my town. This is the White Tower, the emblem of the town on the promenade and the favorable place of citizen of Thessaloniki. And here is the Thessaloniki promenade by night. Thank you very much for your attention. I provoked you to visit this town. It's a very beautiful town.
Thank you very much, Professor
The last,
Athena.
what I want to say,
Yes,
I want to
yes,
make an
please.
announcement that I am a guest editor of special issue Recent Trends of Enzyme Inhibition and Activation in Drug Research and Molecule, and I invite all our participants who can to apply to submit a paper in this issue. Thank you once again.
Thank you very much, Professor Ithana Girankiki, for an excellent presentation on the status of antimicrobials and your studies on them, and having found some active indoles and thiazoles, both against the bacteria as also against the fungi. She has told that bacteria and viral infections have caused number of epidemics and pandemics. Recently there is a decrease in development of antimicrobial agents. She has also mentioned that some bacteria are beneficial, and all of us know that the health of an individual is known by the gut bacteria he has. They determine how good you are, and they are now used even in therapy. And her studies have found some bioactive. She has also done the molecular docking as also bioinformatics to optimize the leads, and have found that 2 or 3 good leads both against multi-drug resistant bacteria as also against some fungi. Congratulations, Dr. Athena. We hope that they become drugs of tomorrow.
Thank you very much.
There is more attention paid to development of inhibitors of the antimicrobes, which are causing havoc all around the world. And disease resistance is developing fast because of their fast multiplication. And even in this case of coronavirus, this multiplies extremely fast. We have found that the Merck drug has already gone out of use because of the fast multiplication of this, and a number of variants are available now. Omicron is causing havoc all around the world, particularly in the Europe, and in Europe is UK and then USA. Developing world is completely safe at the moment as far as the things are concerned with the Omicron, and we hope so that it remains like this. Now all papers are open for discussion. Please address your questions to the 3 presenters, and have a question and name to whom it is addressed. We have the COVID-19 therapeutic agents, then the anti-cancer agents, then the antimicrobials. Any questions, please? I feel that all have understood thoroughly whatever was presented, and there was clear presentations, and there are no questions from the audiences. Thank you very much. I thank profusely the 3 presenters for the excellent presentations.
It means that they understand everything or didn't understand anything.
Yeah, I agree with you. I agree.
We hope they all understand everything.
No, presentations were so lucid.
I'm sure for you.
Presentations were so lucid that most of them would have understood everything.
Thank you very much. Best wishes to Anil.
Yeah. No questions.
Thank you, Chairman, for your kind presentation and for all what you did to help us to present our talk.
Thank you very much for the lucid presentations.
I also would like to thank all the attendees to listen our speech. So I hope they will be informative for them. And if they will have any questions later on, they may ask by emails as well. Okay, thank you.
Exactly.
They can
Exactly,
always
actually.
use the emails to address the
Isim, you-
questions.
Atina, yes.
I say you become more beautiful. What you did?
Thank you. Because of the pandemic.
Bodrum has good influence on you.
Yes, I think so. Yeah, okay.
You escape
So-
from stress.
Yes. It's very nice to see you as Atina, Vladimir, all of
Yes.
you. So it is very
you.
nice to be all
Unfortunately, we
together
have
here.
no opportunity to see each other. I prefer to be in India this time, but unfortunately, it's not possible this period.
Yeah. Maybe
Yeah,
at
yeah,
this
that's-
71st anniversary of Anil, we'll come together. Okay.
Maybe,
yeah.
I hope so, but it's too late. I want to be in India more earlier than next year.
Yeah, you should have come to the earlier.
It will be
Yes.
a conference of Indian Chemical Society in February, in March.
Yeah, okay.
Anil.
Actually, near Kashipur, there's a sanctuary, and I hope you are able to visit that, or the organizers will take you to that sanctuary.
We visited Kashipur together with Isin some years ago, and
Yes,
went to
we have
National
been
Park.
there.
And went to National Park. You remember really how much raining it was without light. We stayed at
Yes.
night. It was nice experience. Very nice.
Yes. With the monkeys. All right.
Manchin.
Yeah.
No, Manchin was not with us. Vafa
Although
was.
you would
Manchin
not have
was
seen
not.
any tiger over there, monkeys you would have seen.
Yes,
Manchin
yes.
was not. Was Vafa. Manchin was not with us.
Hello.
Only Vafa.
on
Unfortunately,
Yeah,
which is
I'm
who
very
is not
sorry.
now with
Hello.
I'm
us.
very sorry. Yeah.
Yeah, I hand it over to the organizers now. Yes, Dr. Saxena?
Yeah, hello. First of all, I thank you, Professor Dr. Kamboj, for chairing this session and for all your support and your wishes. And indeed, it has been a very great pleasure that we could be together. Even if you were not able to come here, but I am feeling as you are here itself. No, so thanks to the IT
That's true.
technology
I am
also.
able to enjoy the
So
whole
I would like
presentation.
to give the thanks and memento to Dr. Kamboj. I don't know how they have arranged this, so this will be sent to you. No problem. Now I think to Professor Vladimir Poraikov. You can put the screen again.
It is on the screen.
Professor Poraikov, actually, I think it has been really a great pleasure with you also. And I was thinking many times that you are very close to us here, and your lecture, particularly on this topic, which is of so great importance of repurposing our drugs for COVID, was really, really fascinating. And I wish you all the best in that further. Thank you so much for your support and for taking time to give this lecture, and for the wishes and all the pictures you had shown. So thank you so much, Vladimir.
Thank you.
Poraikov. Thank you. And this is
Anil.
for you. And then we come to Esin Aki, and Esin, yeah, again, I'm also finding as if you are talking here, you know, when we are meeting and we are together all the time.
Yes.
And this is for you. And again, you had been here, and you must be remembering the changes in Jipar which have taken place when you came in 2010. Now we have a good auditorium and good gathering. And so thank you so much. And we feel that you will be visiting again. And also to Poraikov, you had been there in 2010. So wish you
I
all the
hope so.
best in your works.
Thank you
so
very
much.
much. Thank you very
Yeah.
much.
Now the next one is a very, very good friend of mine, and we had been very close, and we are close even now. And many times we are talking also, Athena. So
Yes.
many thanks to you from my side and from my family as well, and to all of you. And particularly that you had agreed and you had given this talk. And I think some of the compounds will certainly come up, which you have designed as antibacterial and antifungal. So thank you so much, Athena. Yeah. Wish you
Dear
all the
Neil, it was
best.
not possible to miss this event. I would like to say to be with you this special jubilee, to celebrate with you, with Aruni, with
Mridula is here.
Mridula, and with Shruti. But unfortunately, COVID didn't allow us to do this. But with heart,
Yeah,
I'm
that's
here
true.
near to you.
Thank you, Athena. Looking forward that we shall meet here again. Thank you so much
I
for
hope.
all, and thank you so much to all.
Professor Saxena, you had messages from everybody except your wife as your boss at home, Mridula ji.
Huh? Yeah,
You
yeah.
didn't give her a chance to express her feelings.
Thank you. Thank you very much. All.
Thank you,
Sister
Professor
Nandini.
Boroikov. Thank you, Professor Eshineki. Thank you, Professor Athin. And thank you, Professor BP Combo sir, for supporting us. And on behalf of the organizer, we convey our cordial greetings to and best regards to you, and hope see you in the new year 2022, COVID-free 22 in advance. See you. Thank
Okay.
you very much.
Thank you. Thank you very
See
much.
you. Thank you.
Thank
also.
you. Yeah.
Okay.
Bye-bye.
Bye-bye. Bye-bye.
I guess
Bye.
it's finished.
Cecil,
Yes.
yes.
Yes, I think so. Bye-bye. Athena.
Bye.
Ciao. My regards
Yeah.
to Ismail.
Yes,
Yeah.
unfortunately, he's out now. So for that reason, he cannot join me here. So he also says hello to you.
Convey my regards to him.
Okay.
Thank you.
Okay.
Bye-bye.
Thank you very much. Okay. Bye-bye, Professor
Ciao.
Cambo. Ciao.
Bye bye. Bye bye.
Bye bye.
Bye bye, everybody.
Now I would request all the delegates, all the students and delegates, please have a cup of coffee, tea, please. And you have to come back to the seminar hall within 4:30, please, in advance. Maybe within 5 minutes you will have your tea or your coffee, please. Because now this session is for young scholar and researcher. PhD scholar they will present. So for the students it is very important. Within 5 minutes you complete your tea and come back.